So I will be directly responding to Jeff's secondary points. One of his secondary points was that humans and animals are so different that testing is not applicable. According to the Group for the Education of Animal-Related Issues, approximately 95% of all lab animals are rodents, and they are the preferred animal model because, as he said, their physiology and genetic makeup closely resembles that of humans. And he also mentioned that they share 99% of all the genes with humans, and their genomes are also easy to manipulate to replicate the human form, which makes them even more similar to humans. And also, genomic knowledge has made it so that animal research can be much more specifically targeted and accurate when representing a human, thus correctly predicting how a human will react. So even though they might seem extremely different, modern technology has now made it possible so that they will yield the same results. He also mentioned the drug Vioxx. While it is true that it did result in many human fatalities, it is not a case where animal testing results were skewed. It was simply a case where there was not enough testing because it has such incredibly rare side effects that only massive clinical trials could have had any hope in detecting them. According to the Protest Committee, an Oxford-based organization, the number of animals used in the Vioxx trials was extremely limited for ethical reasons. Only a few hundred were used. But this made it impossible to detect the side effects because they were so rare that they showed up only in 1 in every 400 cases. And in order to detect such a side effect, at least 5,000 animals would have been used in order for the data to be statistically significant, rather than just seen as a chance occurrence. Another secondary point of his was that alternative methods should be used in place of animal testing. According to the American Anti-Vivisection Society, increasingly scientists are looking at non-animal models to provide more and more answers. That's not only going to decrease the number of animals used in certain experiments, but more important for many, speed up the drug approval process. One example of an alternative method that's already become the norm is one that I'm sure many are familiar with. Not too long ago, a woman had to buy a laboratory test that involved killing a rabbit if she wanted to find out if she was pregnant. However, now she can simply buy a small over-the-counter kit that tests her urine for certain chemicals, and this shows that rabbits have been completely replaced with an alternative method. And another alternative method that is up and coming, that I'm pretty sure he mentioned, is microdosing. And while this is a viable option, perhaps in the future, it has limitations just like any other method of testing. So there's no guarantee that the human body's reaction to a microdose will be the same as that to a full dose. And although it looks promising, it is, again, a relatively new method and has yet to be fully validated. He also mentioned that cells grown outside the body and computer programs are viable alternative methods. However, according to the Group for the Education of Animal-Related Issues in 2009, cells grown outside a body and computer programs cannot yet predict the complex interactions that occur in an entire living system. Replacement of animals is not here yet, nor is it coming very soon. And according, once again, to the American Anti-Vivisection Society, people in the research community would be the first to tell you they still don't know enough about how the complex living organism works in order to duplicate it. So, for the time being, we don't have a viable replacement for animals, but for now we can use this. We can follow the 3 R's method, which is reduce, refine, and replace, which is basically just reducing the number of animals used in laboratory research, refining the processes in which they are tested, and replacing them eventually with alternative methods. Another one of his subpoints was that most diseases are uniquely human and need human cells, and he mentioned that, for instance, HIV, that it is not exhibited in other animals. Actually, Actually, chimpanzees and other primates have shown similar symptoms in a disease called SIV, simian immunodeficiency virus, which is the primate version of HIV. And so, because of this, rhesus macaques, which is one of the few Asian species who are not endangered and possess no natural defense to SIV, are used in HIV research in order to save more human lives and to slow the progression of HIV to AIDS. And also it has been shown by understanding animal research that many veterinary medicines that are used for the treatment of animal diseases are also used for humans. Examples include antibiotics, painkillers, and tranquilizers. And it's shown that veterinary medicines are very similar to those, or are the same as those used to treat human patients. And it's also been shown that animal research has played a role in many advances in veterinary medicine, as well as human medicine. Because of animal research, they managed to develop vaccines for rabies, the parvovirus, and distemper. And for those of you who don't know, parvo is a very painful disease that many animals suffer, and it includes uncontrolled vomiting and muscle pains, excruciating muscle pains if they are not vaccinated. Not only that, but many treatments don't have to deal with the diseases. It's because of animal testing, they have managed to create pacemakers, hip replacements, diabetes treatments, and dental care, which are used for humans. And the last point is that animal testing is junk science, but that is not true because there's plenty of evidence around that animal testing works. For example, animal testing has led to advances in our knowledge that may help us develop additional cures for malaria, tuberculosis, typhus, and the function of neurons. And it's also been shown that vivisection was crucial in the discovery of anti-blood clotting drugs for the treatment of hemophilia. And many years ago, many people died from simple cuts or nosebleeds because they could not stop bleeding, but now that is not the case. And so, as you can see, while there are alternative methods available, there is just no way to fully replace animals as of yet.